Thank you for coming to Endocrine Seminar, this kind of dark and windy Wednesday afternoon. Hopefully it won't be too bad by the time we got out in about 50 minutes. It's my pleasure to welcome and introduce Dr. Josiah Hardesty. Josiah is an assistant professor in the Department of Pharmacology and Toxicology. He received his bachelor's degree from the University of Kentucky in 2013 and did a Ph.D. in my favorite department, biochemistry and molecular genetics, and completed his degree in 2018. He was a student in Matt Cave's lab in the Division of Hepatology in the Department of Medicine. And during that time, Josiah contributed to 10 papers with 4 as first author. So you can see that Josiah is very hardworking and really a creative and proliferative scientist. He completed 5 years of postdoctoral training in the Alcohol Center with Dr. Irina Kirpchak, during which time he also worked with Matt Cave. And he was awarded an F-32 grant, which is a major achievement from NIEHS. And he published 18 papers as a postdoc. He's presented his research at national and international meetings and received a number of prizes, both for his posters and for his oral presentations. Just a year ago, Josiah joined the Department of Pharmacology and Toxicology as a tenure track assistant professor. And in total, Josiah, despite the, roughly 11 years of his career, has published 38 papers. So he's just so highly productive. It's just amazing. And since starting his lab, Josiah has published 3 papers, including one as a senior author, on which his postdoc, Tyler Gripshover, is the first author. And that was just published last month. So, he's off to a great start in terms of doing all the things necessary for promotion and tenure here at UofL School of Medicine. Josiah is currently mentoring that one postdoc. He's very organized and hardworking. And personally, I know that Josiah is a great collaborator. We have a couple of grant applications in that we're hoping might get funded. Josiah is currently PI on a K99. He's now in the R00 portion of Mosaic grant from NIAAA, restoration and preservation of hepatic cardiolipin levels promote liver regeneration and alcoholic hepatitis. I guess. He was previously PI on an LRP award from NIAA. He's on the editorial boards for 6 journals. I don't know how he sleeps at all, and is serving on the UofL Presidential Scholars Review Committee. So it's a pleasure to welcome Josiah, and looking forward to hearing about the work.
Thank you, Carrie, for that introduction, and thank you for inviting me to kind of share some research I'm excited about, because as Carrie mentioned, I did my doctoral research with Matt Cave, where we were kind of looking at what was MASLD at the time, and now is the name changed to metabolic associated dysfunction, steatotic liver disease. But we're, as a lab, we're kind of getting back into MASLD and doing animal model work and looking at multi-organ gene expression changes in a mouse model of MASLD and looking at the impact of intervention. So I'm curious to see some of your thoughts on our research as a lab. So I have no conflicts of interest to disclose. So today's learning objectives, I want to really just highlight recognize that MASLD and its pervasiveness in society, identify different organ tissues and their respective transcriptomic changes in MASLD, and outline some of the preliminary findings we see from our multi-organ transcriptomic data and the impact of alcohol abstinence and intervention strategies. And I just want to also emphasize the interesting component that there are some gene expression changes in biological process that we see don't improve with some of the intervention strategies we incorporate in our animal models of MASLD. So just a quick outline, just to introduce MASLD, the pathology, the different stages, the current therapies that are available. And I've broken my talk down into 2 different studies. One is kind of characterizing a model of MASLD, looking at 2 variables, a high-fat diet and ethanol, and then a second study looking at the impact of dietary intervention and alcohol abstinence. and then finishing up with some conclusions for those studies. So as I mentioned, the nomenclature for liver disease has changed recently, shifting from non-alcoholic fatty liver disease to MASLD or metabolic dysfunction associated steatotic liver disease. And there's also, it's more of an inclusive term now, and also looks at MASLD and considering the consumption of alcohol as well. Um, and then, of course, some of the more other chronic liver diseases over here, such as ALD, cryptogenic SLD, and drug-induced liver diseases are still here. But I'll be focusing on today MASLD and MET-ALD primarily. But I just want to reintroduce that nomenclature change before we get started. So MASLD has, it can have many modifying factors that I have listed here, but one of the key pathological stages going from top to bottom here is that individuals who have a hypercaloric diet and or also consume alcohol chronically can develop steatotic liver disease or lipid accumulation in the liver is pictured here. And this can progress further to have inflammation within the liver and immune cell infiltration. And then what's concerning clinically is more of the fibrosis and the cirrhosis, and more of the end stages of the liver disease pathology. So in our society, MASLD impacts roughly 30% of the global population. They could fall in any one of these criteria, the pathological stages from simple steatosis all the way up to cirrhosis. And it not only impacts the liver, but has many multiple impact on multiple organ systems, mainly cardiovascular and kidney function and kidney disease, and the healthcare costs roughly 100 billion annually in the U.S. alone. And like I said, there's multiple factors at play, including diet and environmental chemical component, alcohol, sex, and genetic factors. And then here's kind of just a rough, model showing some of the known biological processes that are impacted in MASH and MASLD, such as gut endotoxemia, gut leakiness, lipotoxicity in the liver, inflammation in the liver. And then this can also lead to different impacts in the heart, the cardiovascular system, the brain, and so forth. So currently, the only FDA-approved therapy for MASLD and MASH is resmetirom. And I joke now with my postdoc because when I started as a graduate student, we had a meeting at ASLD, and my mentor, Matt Cave, who was part of a section on nuclear receptor research for treating MASLD at the time. And we're in a room quite like this with a bunch of clinicians, and everyone said, nuclear receptor therapies aren't really going to cure NAFLD. And it was kind of disheartening at the time because all we were doing was looking at nuclear receptors, environmental chemical research, and the impact on NAFLD. But one has shaken out in this THR beta agonist, and it's definitely a starting point. It's not perfect. We're only roughly 30% of patients compared to placebo control see a resolution in MASH with no worsening fibrosis. But I just want to highlight that there's room for improvement and room for new drugs. So for study one, it's focused on the development of an animal model of MASLD. And so what we did for this animal study is we had 6 groups of mice here. The color code here will be consistent throughout the figures, and we had 13 weeks of this animal study. And at week 10, we did our glucose tolerance test. So CD is what we're referring to as the control diet, and here's some of the percentages of the macromolecule in that diet. And then our high-fat diet is pictured here. And so control diet with water only is yellow, control diet with ethanol, this kind of burnt orange, high-fat diet with water, maroon, purple is high-fat diet with ethanol. And the ethanol is 10% ethanol in the drinking water. And then we had 2 additional groups here that had access to both the control diet and the high-fat diet and water or alcohol. And this control diet it does have a higher percent of fiber as compared to the high-fat diet. And I'll kind of touch note on that in some of the findings we found later on in the study. And just briefly, the statistical comparisons made for this study were based on our two variables of diet and ethanol with three data set comparisons, since we have six groups here. And we use a Tukey multiple comparison test. So one, the clear findings was that all the mice that had access to a high-fat diet gained more weight. It was interesting, the high-fat diet ethanol group actually started to wane after about week 6. But as pictured here is the final body weights, where we see a diet effect and also an ethanol effect with a slight decrease amongst the ethanol groups. And our blood cholesterol levels are pictured here and blood LDL levels. So this was interesting in that the high-fat diet with ethanol actually had an exacerbation on the blood lipids, namely cholesterol and LDL. And then our liver triglycerides, as pictured here, were mainly driven by the high-fat diet effect. And this is kind of recapitulated in our H&E staining of the liver, where we see more steatosis, primarily in the mice that had access to high-fat diet. So kind of the takeaway from here was that, going into it, we know high-fat diet's bad, we know alcohol's bad, but really the high-fat diet and ethanol was only exacerbating the dyslipidemia and the cholesterol and LDL that we saw. We then looked at the glucose tolerance test data from week 10, and then the what's pictured here on the far right is the fasting blood glucose levels at the end of the study. And so when we look at the area under the curve for the glucose tolerance test, there was a striking ethanol effect, improving the glucose tolerance, which at first glance we're kind of surprised, but it seemed like in the literature there's a lot of support for that finding, at least in the animal model studies that we see. And then the fasting blood glucose, surprisingly, was higher in the mice that had access to both diets, as pictured here. So, with this in mind, we next wanted to look at some key hormones that may impact some of the gluconeogenic or glucose storage to see what the impact is in each of these experimental groups. And so for this, we looked at plasma insulin, and we really see hyperinsulinemia mainly driven by a high-fat diet, but it's higher in the mice that had access to both the control diet and high-fat diet. The HOMA-IR score, similar trends. And then we also looked at active GLP-1 and plasma glucagon. So just a little background. GLP-1 is cleaved from glucagon. And so we see similarities between both of them pictured side by side here. But what was interesting is that this control diet, high-fat diet group with ethanol actually had a lower active GLP-1, but still had high glucagon. And we're still trying to figure that out. There's been some research looking that GLP-1 can come from the intestine, and we think there may be an intestinal or a microbial component that's influencing these effects here. But and then, of course, the plasma leptin kind of mirrored the body weight changes that we saw, mirroring that. And then PYY over here, we see mainly a high-fat diet effect where it's lower in the high-fat diet-fed mice. So this is some interesting data that we're still trying to kind of work around and see what. Yep.
Yeah, that's really interesting about the GLP-1.
Yeah.
Because GLP-1 is like a carbohydrate sensor, right? Over protein in in the intestine, in the enterocytes. So, yeah, do you have a theory why the mice? I can see it's increased with ethanol, which kind of makes sense because that's a carbohydrate source, right? But the control diet is just doing something.
Yeah, that's something interesting, and we'll kind of yeah. So they have access to both diets, but in this next screen, I'll show you that while they have access to both, they strongly prefer the high-fat diet, where they consume more per gram per week. But we also we do have some 16S data that we're trying to investigate further, but we do see gross changes between some of these groups where the mice that have control diet, high fat diet, and ethanol, they're not reverting back to a control diet mouse, but it's definitely closer than the high fat diet alone. So again, it's interesting data. A lot of this data is going to be, so we just finished this animal model in March of this year. So we're kind of putting everything together with different omic technologies. So we're still preliminary phases, but it's kind of interesting, and I'm open to any input from you all. Yeah.
Endogenous GLP-1 measurement, is that, do you have like a kit or how?
Yeah, so we in our floor we have a mesoscale diagnostics instrument. So this is a panel of six kind of, it's called the metabolic kit. So it has these six hormones that you can measure. And it's pretty cost effective. I think it's like 1800. So instead of like six ELISAs at 500 or 600 piece, it's kind of, and it gets it all in one. And you use roughly the same amount of plasma as you would for one ELISA and one kilo.
It measures GLP-1.
Yeah, so this one, it's I haven't included here, but that's the active GLP-1. They also have one for total as well. Mhm, yeah. And so kind of to my point, these are just kind of the rough, you know, area under the curve for the control diet here, where we see a slight decrease with the ethanol preferring mice where they drink or eat slightly less control diet than the study. Um, and the mice that have access to both control diet and a high fat diet, they eat substantially less, but they're still eating some of it. And then the high fat diet, we see that it's kind of dropping when they have access to both, and even more with the ethanol access. And then the water is less in the high fat diet groups. And so that's something else, probably consistent with the texture of the diet, possibly. And we see similar with the ethanol. And then we went back and kind of calculated the kilocalories per mouse per week. And we see that there's a diet and an ethanol effect where they're consuming more kilocalories per week for those experimental mice, which is maybe contributing to some of the weight gain, at least in these last two groups that we saw. So as I mentioned, we did some preliminary 16S sequencing to see kind of where are the gross changes in the gut microbiome in these experimental mice. And here in the yellow and kind of dark orange, we see clearly the control diet mice have a distinct microbiome as compared to the high-fat diet mice. But you see the high-fat diet alone mice are kind of tucked away in this corner, and the ones that had access to control diet are kind of not directly in the middle, but kind of shifting more. And then when we look at the Bacillota or Firmicutes Bacteroides ratio, we see that it's significantly increased in the high-fat diet ethanol mice compared to the high-fat diet water mice. And it's kind of down in both the control diet ethanol groups. And these are just kind of representative chronodiagrams of the different Bacillota, Bacteroides, and Akkermansia to see kind of the similar to the PCA, some of the shifts in some of these microbes. So we're still kind of investigating this to see what are some of the kind of specific changes that may be contributing, that may be associated with the fiber in the diet if it's shifting the microbial population. So we next want to evaluate liver injury in this model. And so if you looked at all the data beforehand, the last two groups actually, you know, gained the most weight, seemed kind of hyperinsulinemic, had worsened glucose tolerance. So you kind of assume the injury was worse, but actually we see that the injury is better in these groups. And it's kind of peaking in the high fat diet water, high fat diet ethanol groups, at least in regard to ALT, ASTs. And then also we measured plasma and cytokeratin 18 as a necrosis marker. And then pictured to the right is our tunnel positive staining, where the dark brown are the tunnel positive cells. So this is kind of surprising to us, or this is one protective effect we see with the access to both diets. Yep. Yeah.
Giving both, have access to both.
Yeah. Yeah.
Are they mixed together, or are you measuring how much of one are eating versus the other?
Yeah, so we actually, instead of just having one container in a loft, we have both diet, both water containers. So we can have two liquids, two diets, and they're not mixed, they're separate. And then week to week, we change the orientation. So there's not like, if control diet's next to ethanol, they're going to stay away from that just because of the ethanol. So we switch them back and forth kind of every week. But
Diets have the same number of calories.
no, no. So the high fat diet has more kilocalories per gram, 5.49, and 4.17 for the controlled diet.
They show you how that.
Yeah, yes, yeah. And these are kind of just the macromolecule distributions for those. Mhm.
So like if you eat more of the high fat diet, do they get fatter?
Yeah, so that's one thing that we see here. So these two lines at the bottom, these are just the control diet, either control diet water, control diet ethanol mice seen here.
Thank you.
And so this is one of the endpoints that we looked at where we see some protection with the control diet access as well, which was kind of interesting. We next wanted to look at different cytokines in the liver, namely IL-10, which is a pro-resolution factor, and TNF alpha, which is pro-inflammatory. And then we also looked at plasma endotoxin. And so IL-10 was higher with the high-fat diet, but it's even increased further with the mice that have the control diet access as well. Liver TNF was highest in the high-fat diet water group, surprisingly, but higher with just the high-fat diet compared to the control diet. And then our endotoxemia was primarily driven by a high-fat diet as well. So I know I showed kind of a lot of data, a lot of different groups, but kind of the synopsis is up here where just the high fat diet effects are pretty apparent and consistent with many of the other studies where we see increased obesity, steatosis, reduced glucose tolerance, gut dysbiosis, liver injury, liver inflammation, endotoxemia. The exacerbation between high fat diet and ethanol seemed to drive the dyslipidemia we saw in that first slide. And then ethanol alone was improving glucose tolerance and active GLP-1 and glucagon. And the control diet, high-fat diet, was reducing glucose tolerance, leading to hyperinsulinemia, reduced active GLP, and reduced liver injury and increased the IL-10. So kind of the take-home points from the study was it seemed like high-fat diet was the main driver of the MASLD phenotype. And it hurts me to say that as being an alcohol researcher now, but it seems like the high-fat diet was kind of winning out in that effect. But we did see some exacerbation with ethanol. And again, we see some surprising effect on the glucose tolerance test with ethanol alone. And with the mice that had access to both diets, they appear to have worsened diabetes, but improved gut microbiome profile, reduced liver injury, and increased liver IL-10, pro-resolution cytokine. And this may highlight the importance of dietary fiber and MASLD. So currently we have RNA-seq data from these experimental mice, and we're trying to kind of parse apart different transcription factors that may be activated or inhibited based on the variables we're looking at. So we're exploring that further for this study. Uh, we're also we're trying to understand the impact of the dietary fiber because one thing that we see in the preliminary RNA-seq data we have is that a lot of the Kupffer cell gene markers are upregulated in the control diet, high-fat diet fed mice, but they're going down pretty dramatically with ethanol in the high-fat diet group. So we know they're important for the immune system and the liver. So we're trying to understand, is this purely kind of a gut liver axis story, or is there something going on within the liver that's influencing these important immune cell populations?
Okay. Do the lipid fractions in mice, do they function similar to those in rats? That is, that HDL is the main carrier?
That's a great question. I'm actually, a lot of my research hasn't crossed back into the rat mouse comparison, so I'm not 100% sure on that. Which one's the main carrier for the lipids between the mouse, or at least in the rodents between mouse and rat? that could be something we could look into to see if anything's maybe indicative.
Because that's different from rabbits in the original study
Mhm.
done with high cholesterol done with
Oh.
rabbits, and they never repeated
Hmm.
rats. But I don't know about mice.
Sure. Okay. And so this next study is kind of looking at more of a not necessarily a therapeutic intervention, but just a dietary intervention where similar, we had a 13-week study, and we had the cohorts of mice that were all fed a high-fat diet for the first 7 weeks, and then one cohort was switched to that control diet that I described earlier. And the what I'll refer to as the full choice group had access to control diet, high-fat diet, water, 10% alcohol, and the intervention group was the same for the first 7 weeks, but then switched to where they only had access to the control diet and water. And so this is kind of like a common sense thing where, we kind of knew going into it that, giving mice only access to control diet should alleviate a lot of pathology. But what we're really interested in is when, a mouse basically is restricted to where they're making the healthy choice every time they only have access to control diet and water, what reverts back to normal, and then what changes actually don't change with dietary intervention or alcohol abstinence. And maybe that's what we want to therapeutically target in our next form of studies. So for this part of the study, we're using an unpaired student t-test to compare these two groups, the full choice and the intervention group. So here's the body weight throughout the study. So around, when we switch after week 6, week 7, we see that the body weight's starting to drop back down. As we see here with the pink with the intervention group as opposed to the full choice. Let's recapitulate the final body weights, and then similarly, the white adipose tissue weight is down, the blood cholesterol levels are down. We see the liver triglycerides are reduced, blood LDL and VLDL are reduced as well, and we see the steatosis is improved in the intervention group. So, this is kind of upfront, we kind of expected this, so it's kind of reassuring. We next want to look at the glucose tolerance between these two groups. We see improved glucose tolerance in the intervention, as pictured here. Fasting blood glucose is down at the end of the study as well. We see a reduction in plasma glucagon, insulin, leptin, and PYY wasn't really changed. We'd almost expect it to go higher with the intervention, but it didn't really have a change. And then the HOMA-IR score was improved as well with intervention. We next similarly looked at the 16S metagenomics data. So we have our 4 intervention groups here, and then our full choice groups over here, showing separation by PCA analysis. Then we see some kind of contrasting changes between the groups here on this level, taxonomical level. So, we're seeing changes in the gut microbiome populations, improvements in the glucose tolerance, and also the kind of metabolic syndrome endpoints as well. So we next want to look at liver injury and see improvements with intervention, ALTs, ASTs, plasma cytokeratin 18 is a necrosis marker as well. And we see a non-significant decrease in the endotoxin, significant increases in pro-inflammatory cytokines, IL-1 beta, KC-GRO, and interferon gamma as well, suggesting liver inflammation is reduced with the mice that received the intervention. So, clearly it is pretty unanimous that the dietary intervention, alcohol abstinence, clearly improved MASLD at all our kind of phenotypic markers. So now the next question is like, what do these lifestyle changes not improve? So what, you're both consuming high fat diet predominantly with access to 10% ethanol, and the mice that received the intervention, what's not changing compared to mice that just had control diet and water the whole time? And can we use this information for the development of novel drugs that, clearly if lifestyle changes improve something that's great, but what even if you improve your lifestyle, don't change. Maybe that's what we want to target. So with this, we wanted to do multi-organ RNA-seq from our control diet plus water mice. So for the full 13 weeks, they only had access to control diet and water, kind of our healthy group, the full choice group, which is kind of our unhealthy group, and then our intervention group here in yellow. So we have liver RNA-seq data, brain RNA-seq, white adipose tissue, ileum, and skeletal muscle RNA-seq data from these three groups of mice. And what we're interested in is with intervention, what is still significantly different from the control group. And so this, again, very preliminary. So I'm just kind of went through multiple tissues and kind of highlighted some interesting gene changes. But we hope to kind of do a deep dive into this data to really see what kind of therapies we may want to pursue for the next type of preclinical animal study. So for the hepatic RNA-seq data, we see roughly 215 genes that are differentially upregulated in the intervention group versus the control diet water group, and 139 that are decreased. And since this is kind of a endocrine theme talk, I felt that these two genes were pretty interesting. So these are lipases, one being the hormone sensitive lipase E pictured here, where in the full choice group, it's the highest expression. Intervention it reduces a little bit non-significantly, but still significantly higher than the control diet water group. And then the other lipase C here is highest in the control diet water group as compared to the intervention. So this just kind of highlights that there are some gene changes that we're going to follow up on to see what is the biological meaning and why they're not, kind of self-correcting with lifestyle changes in this MASLD model. Next, we looked at the white adipose tissue from the EpiFat and looked at the kind of gross differential expressed genes as pictured here by a volcano plot. And one that was of interest is CD276, which did go down pretty substantially with intervention, but it's still higher than the control diet water group. And this has been shown to be important for some of the inflammation of the white adipose tissue. So it being higher could be contributing to inflammation in the white adipose tissue, even after the lifestyle intervention. Next, we looked at the skeletal muscle and the differentially expressed genes. So comparatively to all the other tissues, it had the fewest amount of genes that were differentially expressed, which may be expected, maybe compared to liver and fat. But what was interesting was the leptin receptor was highest in the control diet water group, but still downregulated in the intervention, almost to the full choice levels pictured here. Yes.
So it looks like it's very low expression if I'm reading that.
Yeah, the TPM. Yeah.
Interesting.
Yeah, so the TPM, similar to the predecessor at PKM and RPM, it's a kind of a normalization unit for the RNA-seq data. But yeah, it is lower, it appears in the skeletal muscle. And similar, so this is taken from the endocrine panel that we looked at with the plasma leptin levels, so just showing kind of the contrast where the plasma leptin levels are lower with intervention. So that's one thing the intervention properly did correct. The leptin receptor is still pretty low. And then our gene expression changes in the ileum. One is the ApoB receptor, which is some interest to Carrie. I thought it was interesting that it's higher in the control diet water group. Yeah. And the intervention group didn't raise it substantially, and it's still kind of at the full choice group level, which is interesting. And then, interestingly, the brain had the most amount of gene changes, which it's kind of a new area for us as a lab looking at the brain. And so we haven't done too much of a deep dive, but we do know that the brain barrier, blood-brain barrier, is very important. And one of the genes that was still low in the intervention as compared to the control diet water was this tight junction protein 2 gene, which is important for it's one of the important proteins for the maintenance of the blood-brain barrier, but that was kind of interesting. So, in summary, we see that, basically, healthy diets and alcohol abstinence kind of essentially, cured the MASLD phenotype we saw. But what we're, really intrigued by is seeing what it didn't improve with the lifestyle change and how we can use that information to maybe make more targeted therapeutics to test in the preclinical animal model setting for MASLD. So future directions, we want to kind of do a more thorough investigation of the RNA-seq data that we do have from the multi-organ tissues that we have from this animal study, and then move on to develop and assess therapeutics that may be more efficacious for MASLD. We want to try to avoid therapies that are improved by lifestyle change because, there may not be an additive effect. If the lifestyle change is the predominant factor, we want to kind of see things that don't change with the lifestyle intervention where that we may be able to capitalize more on improving the phenotype. We also want to look at exercise as an intervention with treadmill studies as well and do something similar in a MASLD model. So with that, I'd like to acknowledge the members of the lab, postdoc Tyler Gripshover, my lab technician Rui Treves, and then Dr. Craig McLean, and then NIH for the funding for the R00. And that I'll take any questions. And then today's event code is listed here.
There's so much data and so many interesting leads to pursue that I'm really impressed.
Thank you.
Yeah. And the idea of looking at multi system communication, I really like that.
Yeah, thank you. Yeah, I didn't touch base on it, but I probably should have. But we also we have a way that we can identify at least at the gene level what's a predicted secreted protein, so we can actually look at the organ crosstalk and see, what hormones going down here, and is it actually changing the plasma and measure it in the plasma as well. That's something we're trying to pursue to fit all the pieces together. Yeah, it was.
Especially with dementia.
Yeah.
It'd be interesting to look at those aspects.
Yeah,
Insulin resistance, like GLP-1.
Yeah.
The question there
Oh.
on the chat.
Okay, so the question in the chat is, was the brain RNA-seq in the last study on the whole brain or a particular part? So unfortunately, it was on the whole brain. We're kind of liver people, so we're not very sophisticated, but we are interested in, it's kind of an offshoot of this experiment, but the full choice group, we also measured phosphatidylethanol in the blood. So that's a marker of chronic alcohol consumption. So we separated that group into the mice that drank more alcohol and drank less. And we're interested in doing a follow-up study with brain spatial transcriptomics. And we're learning a lot about it. We're interested in the slice where we can look at the nuclear accumbens and the areas of the brain related to addiction. But it's definitely a key point that this was the whole brain and not a specific area of the brain.
Initially you said about the receptors and how the thyroid hormone has found to be having an effect of brain studies so many interact with thyroid hormone, alcohol.
Yeah, so that's a good point. So that's we haven't actually found anything conclusive, but since we have primarily transcriptomic data, we're kind of interested in looking at transcription factors and get a kind of a proxy measure of activity. And similar to what you were mentioning, see if there's any crosstalk with thyroid receptor beta, for example, to see maybe there's a natural improvement of signal just with, dietary intervention and alcohol abstinence, but we haven't saw anything conclusive yet, but we're in the process of looking at that.
Well, yeah, now I was under the impression that, at least in human beings, the major risk factor for the metabolic liver disease is actually too much fat. So, now, so I'm wondering, can you differentiate how the diet plays on it? You see what I mean?
Just the total amount of fat in the
Yeah,
livers.
because, my understanding is that, the fat tissue becomes more inflammatory
Mhm.
and that releases all kinds of things.
Yeah.
Leads to insulin resistance and, changes in the livers, the way the switches on and off, the way the liver develops lipoproteins, and, and so on. So, it seems like if you're feeding the rat various things,
Mhm.
is it directly causing some of this to some degree, or is it acting to increasing the amount of fat tissue?
Yeah, because we kind of see them both together, so we haven't really parsed it out, which one's coming first, the white adipose tissue accumulation of the fat or the fat that we see in the liver. And it's kind of interesting. So coming from my postdoc where, we look at the very end stages of liver disease where actually the hepatocytes are not normal and you actually see less steatosis in like a cirrhotic liver because the hepatocytes can't function properly to take up, fat. And a lot of the patients are, malnourished and they don't have a lot of fat. So, yeah, we haven't really explored other than one lipid molecule we're of interest in for the R00 that I'm also trying to look to translate into MASLD is cardiolipin, which is a phospholipid that's primarily in the mitochondria. And there has been data showing that those levels go down, which may in turn lead to less fat burning in the liver, possibly, and mitochondrial dysfunction. But yeah, we haven't really quite parsed out what the chicken or the egg. Is it the fat accumulation, the white adipose tissue first, or the liver that's contributing to the worsening of the metabolic syndrome? Yeah, well, so let me pull back up my slides. Yeah, so that's a good point. So in the liver, we clearly see improvement of steatosis, but that's a good point. Maybe in other tissues, 6 weeks isn't long enough to really see physiological endpoint changes. And so, but definitely just in the gut microbiome in the liver, we do see some physiological changes. So here, this with the steatosis, but that's a valid point. Maybe for skeletal muscle, the tissue where we saw the least amount of gene changes, maybe skeletal muscle wasn't really impacted to begin with, or maybe it would need longer or something. Mhm. Yeah, possibly. Obviously it's what they're, consuming every day, and if they're kind of protein deficient, so to speak, that could be a player as well. So not to say that all the effects that we're seeing were the fiber effects, but that was just one thing that we noticed where in one diet we clearly have some fiber, and the other we have completely absent of fiber. But that's a good point. I think there was one more question in the chat. Uh, So this question is Ahmad Kulkarni. It'd be interesting to check high fat diet plus high fiber diet impact on various endocrine parameters. Enjoyed the presentation. Looked like keto diet offers benefit. So more of a comment than a question.
Yeah, thank you.